The next talk is from Ed Lein from the Allen Institute for Brain Sciences, and Ed's been a leader in the charge that the Institute has for developing these comprehensive transcriptome maps that Alan had talked to you about a little bit earlier. Ed will talk a bit about that, but he's also done a great job together with the people on his team in not just developing these large data resources, but really also gleaning from them some key biological insights about things like cross-species differences and similarities, issues of cell type specificity and regionality in the brain. And so Ed will give us a talk both about the resources that the Allen has developed and some of these new insights.
Well, thank you very much for the kind introduction and for the introduction to speak today. It's really a pleasure. As Alan mentioned at the outset, for almost the past decade now, the Allen Institute for Brain Science has been engaged in creating these very large-scale gene expression atlases as open access resources to the research community. In addition to being sort of these monolithic data sets that one can mine, we assume that the gene expression underlies many of the functional properties of the cells in the nervous system, and that by studying these, we can learn something about brain structure and function. What I'd really like to spend this session on, rather than describing the resources, is to spend most of the time trying to relate some of our efforts to try to uncover the molecular logic underlying gene expression as it relates to brain structure. So to do this, I'm going to focus the talk on three. It's not advancing. There we go. Okay. On three very broad and I think still very open questions, which I don't intend to actually answer, but I'll try to address a little bit using some of our data sets. The first is simply, what is the relationship between the molecular architecture of the brain and the cellular and functional architecture of the brain? And can we identify some principles which seem to explain a lot of the variation that's observed across structures? Second, which is also a favorite topic of certain others in the audience, Nat Heinz in particular, is really can we make some progress towards understanding neuronal diversity? Or in other words, how many cortical cell types are there? Can we sort of start to use these transcriptional profiles to begin to build a molecular taxonomy of cell types? And then third, as we've begun to look across species, we can begin to ask questions about how the transcriptome varies between species. And in particular, I think it's of great interest is what is unique about the human brain. So I'm going to focus this talk entirely on the neocortex, although I could have given this talk on virtually any structure in the brain. But this is a good place to focus. And let me first begin by describing the various atlases that we have at our disposal to begin to address these kinds of questions. So the first atlas that we created was the Allen Mouse Brain Atlas, which is in the adult mouse. And the goal of this was really to try to provide a comprehensive cellular resolution characterization of all genes in the mouse brain. Sort of very true to our approaches to this, we've tried to standardize the process and really scale up to an industrial scale the generation of these kinds of data, in this case in situ hybridization data, to the point that we could look at all genes in all structures. This type of approach works very well for small brains. We've been able to apply this to the developing mouse brain as well. But I think you can appreciate the differences in scale here that the same type of approach would not work very well in the human brain with about 1,000 times the volume and increased complexity. And so in the last couple of years, we've really begun to focus on looking at primate brains, including a project on the adult human brain, and two that are funded by NIH now, looking at the development of the non-human primate, rhesus macaque, as well as the development of the human through a stimulus project involving several collaborators as well. And these latter approaches then have had to take a different approach to try to provide this comprehensive transcriptome analysis while covering as much anatomical space as possible. And that's been done really by moving towards a sample isolation in a small number of specimens, where we try to do a very fastidious isolation of brain regions that can be defined on the basis of Nissl and marker stains, and then do transcriptome analysis using either DNA microarrays or RNA sequencing to get this sort of transcriptome coverage. So taken as a whole, I now sort of think of these really as a transcriptional atlas of mammalian brain development as opposed to a single set of atlases, because there's the ability now to look across age, to look across structure, and to look across species as you use these databases, going across databases. So I'm going to now begin to sort of burrow down in levels of resolution, looking at the structure of the neocortex and the relationship of gene expression to it. So this is a picture of the rhesus macaque adult brain through a coronal section, which contains primary visual cortex, that I really like because it illustrates the two primary axes of organization of the cortex, namely the laminar axis, and the genes have been shown by many people, including ourselves, to very selectively label the different layers, as well as the aerial axis. This particular gene called synaptotagmin 6 does a wonderful job of selectively labeling layer 6 of primary visual cortex that we had identified through transcriptional profiling. So the first question that we can ask is, what is the relationship of the transcriptional profiles of these different components to one another? And to get at that, we took the approach of laser microdissection of individual layers from individual parts of the cortex. We can now compare their expression profiles to one another. And remarkably, the sort of spatial topography, the spatial relationships that I just showed you for layer and area come out very naturally from an analysis of this data. So what you're seeing here is a principal component analysis of these different laminar and aerial samples across the macaque cortex. And I think what's sort of easy to see at a glance is that along the first principal component, you have genes, these samples basically are organized by layer. And not only are they organized by layer, but they're organized in the correct order of layers. So this is a completely unbiased sort of analysis, and the laminar ordering pops right out. The orthogonal axis along the next principal component is areas moving in a rostral to caudal sort of dimension. So kind of a remarkable feature here is that the structure of the cortex came right out from an analysis of the data set at this sort of level. Now, my particular bent as a developmental biologist is to explain this in terms of the developmental origin. So neighboring areas are basically derived from neighboring parts of the developing neuroepithelium, whereas neighboring layers, these are derived from an increasingly lineage-restricted precursor pool, so that you would be most similar to your sibling, basically, to your brother or sister in time. But the broader feature here really is that the spatial organization is reflected in transcriptional similarities across the cortex. Now, an even more dramatic example of that comes from analysis of the adult human brain, which we just published last week, actually, where we had the luxury of being able to assay transcriptional similarities across the entire adult human neocortex. So in this particular project, we did microarray analysis of about 900 different samples per brain, about 600 of which, or 500 of which, came from the neocortex of both hemispheres. And as part of this project, we were able to both profile these and then map the sample locations back into the MR space of that very brain. So we can do a calculation between sort of transcriptional similarity and physical proximity. And on the right-hand side here is a multidimensional scaling plot showing the similarities in transcriptional space between all of these different samples that we took across the adult human brain. And what I think that you'll appreciate right away is that there's a lot of clustering going on here, such that all of the samples from each lobe are grouped together. In red is the frontal lobe, green is the temporal lobe, parietal lobe, occipital lobe. Furthermore, the relationships are the same as they are in the in vivo brain. And if you dive down a level into looking at within a particular lobe, you see that the topography within that lobe is essentially recapitulated in the transcriptional similarities. So for instance, if you look at the temporal lobe, starting at the temporal pole, moving to superior, middle, inferior temporal gyrus to Heschel's gyrus, it's the same as you see in the in vivo brain. Other cases like this, the precentral gyrus is right next to this postcentral gyrus. So in other words, proximal regions of the cortex are most similar to one another, and there's an enormous amount of positional information in the transcriptome of the cortex as a whole. Up here in the inset plot is basically you can actually calculate this, and you can see a near linear relationship between the transcriptional similarity and the actual physical distance of these samples. So how to explain this? This sort of graded expression is very reminiscent of the gradients of gene expression that are described in development of the cortex early on, such as in the mouse cortex. And in fact, we have done a lot of profiling of the developing cortex as well. As part of this BrainSpan stimulus project, we've also had a chance to do some exhaustive profiling of the developing human fetal cortex. And the basic strategy was to section an entire hemisphere and then sample both the layers and the areas across the cortex in a very comprehensive sort of fashion. And what that allows one to do then is to look for genes that have gradients across the cortex. So this is actually a 21 post-conceptual week brain. This is a little bit late in development, but nevertheless, we're able to identify sets of genes that vary nearly linearly from the rostral to the caudal pole, including some very well-known genes that have been described as patterning genes in the mouse. So for example, FGFR3 has been described in the mouse as having a caudal high to rostral low gradient, which you can see here in the 15.5 mouse. And we see the same sort of thing here in the fetal human brain. So it would appear that this sort of graded patterning that has been very well described in mouse development is recapitulated in the human, and there's an imprint of this that still remains in the adult. I think it's worth also mentioning some of the things which we did not see. We were not able to identify lateralization. We did not find evidence for a large number of highly discrete areas, such as one might expect if you were to see distinct Brodmann type areas or other functional areas. So I think the question of the relationship of gene expression to cytoarchitectonic or functional architecture is still very open at this point. So I'm going to step down a level now to the question about cell types that I posed. So when we first started doing the mouse brain atlas, this seemed like an incredible treasure trove of genes from which we could derive markers for particular cell populations. One particular goal of this was to be able to use that information to generate transgenic mice to allow one to target those selective cell populations, which is actually very much ongoing at the Institute at this point. So we basically were able to mine this entire 20,000 gene data set to find those genes which had the greatest cellular specificity, and that was very successful. So for example, in layer 2-3 of barrel cortex, you can find a whole set of genes that's relatively restricted to layer 2-3, but what you immediately start to see is a great deal of variation among these patterns. So some of these genes are throughout the entire depth of 2-3, some are really quite restricted just to, say, layer 2, others are in the deeper part, perhaps layer 3, and this turns out to be the case for each of the layers, that there's a big diversity of patterns that you see from relatively promiscuous of most of the cells to much less dense populations, superficial populations, deep populations. Same is true of layer 6. And the only layer we don't see sublaminarity is 6b because it's so narrow, but you can see a great difference in the density of cells for some of these genes. So this is a little perplexing. There are a lot of different kinds of patterns. And so we were trying to identify those genes which might be labeling distinct cell populations on the basis of this. To begin to approach this, Amy Bernard developed a technique for doing double fluorescent in situ hybridization with the concept that if we could identify genes that have mutually exclusive patterns of expression, these were likely to be functionally distinct cell populations that were being labeled. This type of approach has been very fruitful in the field of GABAergic interneurons in the past. In fact, we recapitulated that pattern. So for instance, parvalbumin, somatostatin, and VIP, these canonical markers are relatively non-overlapping with one another. This turns out to be the case as well with pyramidal neurons. So for layer 5 neurons, Amy was able to identify a set of genes that are relatively non-overlapping with one another. Sort of to preface this, these end up corresponding to different projection neuron types, it turns out. But a point I wanted to make on this is that although we can identify these genes, the patterns that we see with a much larger set of genes look something like this. This is obviously not completely accurate based on pairwise comparisons, but what you see is grouping into major classes, but then it becomes sort of a very big combinatorial set of mess, I guess. And so it's not really clear where in here would be a cell type or a reproducible pattern. So it's clear that pairwise comparison is not going to do the trick. We need to move to higher order comparisons. And along those lines, we've been collaborating with Jean Rossier and his colleagues in Paris to try to combine single-cell PCR with electrophysiological characterization and morphological reconstruction of neurons in primary somatosensory cortex of mouse. And the promising feature of this, this is just sort of a very high-level overview, is that purely on the basis of the molecular profiles, we're able to derive a meaningful set of clusters compared to what's known from the literature and from developmental lineage. So first of all, we can easily differentiate GABAergic from glutamatergic cells, or all bets would be off, I suppose. But within the GABAergic population, these basic clustering methods are able to tease out a parvalbumin population, a somatostatin population, a VIP positive population, and a related VIP negative MPY positive population, which are probably the neuroglial cells. So the point is that basically on a small set of genes, that one is able to sort of derive a molecular classification of cells, which I think is very promising for future efforts where we can increase the number of genes that go into this and do this at sort of a larger scale. We took a slightly different tack to try to look at the excitatory neurons. This is sort of a big summary figure created by Stacy Sorensen that is worth spending a little bit of time on, because what she did was to combine expression of these various marker genes with target specificity by co-labeling, retrograde labeling from various target zones with expression of particular genes. And what she found is two things. The first is that there's definite specificity for any given gene. So many of these genes selectively label populations that are projecting to particular zones. So for instance, one set of these genes selectively labeled cortical-fugal populations, another selectively labeled just cortical-cortical, many labeled subpopulations of both. But if you sort of look at the scope here, there are many different patterns. And the molecular specificity of these suggests that these are each sort of an independent cell population or combination of cell populations. We can't differentiate one from many. And that overall, we really should be considering a diversity of GABAergic types, or I'm sorry, of excitatory types on the basis of their target specificity. This is usually something which is lost in studies of this type because when it's looking in local circuits, whereas we really have to look for the extrinsic connectivity for this. And I think really this would be very consistent with other studies from Justelab and others that show that a lot of the specificity of these types is in their projections, it's in their connections and their axonal targeting. This seems to be the case for these as well. And I should mention that we see the same things as well for the other cortical layers too, for layer 2, 3, and layer 6. So now finally, I'd like to end on the phyletic comparison. It's sort of a glib title here, the primates are not mice. We've had the opportunity to compare patterns between species for many different genes. And in many cases, there are dramatic differences between these genes. So on the top here is a particularly robust example of a gene prodynorphin, which is a pre-pro-opioid protein, which has a class shift between mice and primates. In mice, this gene labels a subpopulation of GABAergic interneurons in the cortex, whereas in both rhesus macaque and human, this is labeling a highly laminar excitatory population in layer 4 and layer 4B, layer 4C beta and layer 4B. So this is just a single example, but it's an example that it's important to have the human information because it can be frequently very different. My colleague Honghui Zheng has recently published a much more exhaustive comparison between species, looking at in situ hybridization patterns in the human visual and temporal cortex. And this is sort of the take-home message here that she identifies between 20 and 30% of these genes that show either significant level, laminar, or aerial differences between human and mouse. This is an astonishing number. This is a very large number that I think really highlights the value of having this other data when considering what a gene might be doing in a human compared to what it might be doing in a mouse. That's a very large number. So now, Corey already showed half of my slide. Thank you for not showing the punchline, actually. We've begun to identify some genes that actually have human-specific patterns among the species we've assayed. We just published on this particular result, which is a differential expression pattern for calbindin-1. This gene we've known, it's a very famous gene, we've known for a long time has a pattern where it's in the dentate gyrus of the hippocampus, as well as CA1 and CA2 of the hippocampus. In rhesus macaque, it turns out to have a very, very similar pattern to that, CA1, CA2 in the dentate, whereas when we look in the human, the pattern is restricted just to the dentate gyrus. This is a single example. It's not quite clear what the functional salience of this difference is, but this is sure to be one of hundreds or thousands of these examples of differences in patterning. And this is the type of difference in patterning that we see even among mouse strains. So I think this is going to be quite common. And a key question is going to be how to sort through this information and understand what the functional relevance of these differences is through creative ways. So let me just wrap this up by sort of summarizing some of the things that I talked about for these three questions. So first of all, there are clear relationships between the sort of transcriptional architecture and the cellular architecture. In my opinion, there's probably a developmental lineage imprint that you see in many of these sorts of patterns. It's taken at this sort of broad scale. But it's really still open how this relates to cortical functional architecture, the sort that Jack Galland was talking about, or that one sees with imaging. We don't see certain elements which are very prominent from a functional point of view. Why not? Are we not looking in the right way? Are these things that you only see in certain cells versus a volume issue? Is this something that happened in development and doesn't have an adult correlate? So it's really not clear. I think from a molecular taxonomy point of view, we're really seeing that there's very strong promise for being able to use these transcriptional profiles as a way of stratifying and classifying types, not to mention being able to manipulate them. But it's clear that we really need new methods for going for single-cell transcriptomics. And then finally, related to the species differences, we need to move from the descriptive to the testing. And whether this is creative mouse transgenics with humanized versions of things or working with stem cells, as Ricardo was talking about, I'm not sure. But we need a way to move into understanding what it is that makes human beings unique. And with that, I'll end. It's really not possible to cite all the various people. We have an amazing multidisciplinary team of people at the Allen Institute that has created all of these resources. These range from informatics to neurobiologists to physicists to engineers, a very broad set of people that all come together to make this happen. This is the only way it can work. Various other collaborators, and I'd like to cite as well Paul and Allen, as well as the NIH and NIH Blueprint for funding some of this other work. Thank you very much.